In the last video we looked at carbohydrates. Carbohydrates were obviously polymers of the single repeating unit, which was called a monosaccharide. Glucose being the most important as far as we're concerned. Plants use it to make starch in photosynthesis, and we use glucose when we store it as glycogen in the body. Now, the monosaccharide in a protein is called an amino acid. It's called an amino acid because it has an amino group and a carboxylic acid group. The amino group will always be attached to the C next to the COH. This is called the alpha carbon. And on that same carbon will be an R group. Now, R could be anything from the simplest amino acid glycine, which has simply a hydrogen there, to more complex ones. In your data book, towards the end of the data book, you are given 2 pages of amino acids, because in us, there are 20 amino acids in total. Okay? So, when amino acids link together to make proteins, the amino group of one amino acid joins with the carboxylic acid group of another amino acid. Now, if you can just cast your minds back to the pathways we did, almost at the very end, we met reactions of carboxylic acids where when they react with amines, they become amides. In this particular case, we are going to form amide links over and over and over again. If you can imagine putting another carboxylic acid here, then the carboxylic acid of one, let's just show you. So let's put a COOH here, and let's finish it off and make the amino acid NH2 with R like that. And then what happens basically is the carboxylic acid loses its OH. You saw this in a previous video when we talked about condensation polymers. So that goes from the carboxylic acid. The NH2 loses one of its hydrogens, and then you end up with a CO, which is a C double bond O, an NH, and then the rest of these molecules. And this link will appear now again and again and again when amino acids join together to make a protein molecule. They're called amide links. Biologists tend to call them peptide links, and it's perfectly okay to call them peptides here as well. If you link just 2 amino acids together, you make a dipeptide. 3 would give you a tripeptide, and then eventually polypeptides. And where does polypeptide change into protein? It's just a big polypeptide. In terms of proteins, you need to know there are 4 types of structure a protein can have. Now, proteins will have, first of all, a primary structure, which is simply the sequence of the amino acids present. Now, if you can imagine 20 different amino acids, which could be hundreds or thousands or tens of thousands of units long, you can see there are a lot of possible proteins. And in us, proteins are probably one of the most important of the big molecules. For example, our skin, our hair, our nails, our muscles, our organs, our hormones, our antibodies, our enzymes, they're all proteins. So proteins feature massively in living systems. Anyway, coming back to the structures. So the sequence of the amino acids is called the primary structure. That sequence will have COHs and or groups like this all the way through. And of course we have a very polar grouping there. NH would give us a very polar grouping. Hydrogen bonding obviously is something that can take place there. We've also got a C double bond O, which is also very polar. And a lot of these R groups are polar as well. And what can happen is that the main sequence, the primary structure, can actually join to itself every so often and form the secondary structure. Now, there are 2 types of secondary structure. Firstly, you can get a kind of spiral, which is called an α-helix, and they tend to have every 3rd or 4th amino acid linking to a previous one through hydrogen bonds. You can also get a kind of β-pleated sheet, which is like a zigzag. And again, hydrogen bonds hold that together as well. So the secondary structure is an α-helix or a β-pleated sheet, and hydrogen bonds hold it in that conformation. Further folding of that same α-helix or β-pleated sheet can produce the tertiary structure. Now, tertiary structures can have pretty much any kind of interactions, putting them together in their particular shape. Hydrogen bonds, dispersion forces, dipole-dipole attractions. You can even get covalent bonds and ionic bonds, depending on obviously what's in this R group. Certainly if there's a sulfur in that R group, then you can get something called a disulfide bridge, which is a covalent link, and it's a very, very strong link indeed. Now that tertiary structure is the bulk of the proteins in us. So they make up basically all of the enzymes and the muscles and the organs and so on and so on. It's very unusual you get the fourth type of structure, which is when several tertiary proteins link together and form a quaternary structure. These would be very complex proteins, things like DNA polymerase, the enzyme that's used in making DNA, hemoglobin, which obviously transports oxygen around the body in the blood. They would be the rare ones. The vast majority of proteins are tertiary structures. You're also expected to know how enzymes work. Nothing too complex, just the fact that their shapes, particularly the shape of the active site, is vital to metabolizing whatever that enzyme is set out to do. So the substrate will come along, it will fit into the active site, and then what happens is the bonds within that substrate are weakened because of the attractions within the active site. The weakening of the bonds lowers the activation energy barrier, making it easier for that molecule to break apart. So the enzyme grabs the substrate, breaks the substrate up, the substrate leaves as the metabolized products, and then the active site is kept free for the next substrate molecule to arrive.